If you happen to follow your local environmental news, there's probably a good chance that you've heard of a group of chemicals called perfluorinated alkylated substances, or PFAS. These PFAS chemicals are man-made organofluorine compounds. Consider the PFAS chemical perfluorooctane sulfonate, or PFOS. This molecule is a chain of 8 carbon atoms, with a sulfonate group on the end. So you have a chain of 9 atoms, 8 of them carbon, with a sulfur atom on the end, and this sulfur atom is bound to 3 oxygen atoms, which makes this particular region of the molecule very electronegative, hydrophilic, and reactive, able to bind or interact readily with other chemicals. Every carbon in the PFAS molecule has the rest of its valence electrons shared with fluorine atoms, and this is where the PFAS chemicals get their name. They're perfluorinated. This chemical feature with the fluorine is really important. The carbon-fluorine bond is extremely strong. It's so strong and requires so much energy to break that virtually no organism, be it a fungus, an animal, or a microbe, actually possesses enzymes that are capable of breaking it. The carbon-fluorine bond is actually the strongest covalent bond in all of known organic chemistry. These PFAS chemicals are man-made, and only man can break these carbon-fluorine bonds. Although we can't do it in our own bodies, we can't digest it, we can only do it in very sophisticated laboratories. And even there, the process is extremely energy-intensive, as it requires very high heat and high pressure to break this carbon-fluorine bond. Because this bond is damn near impossible to break, when PFAS compounds make it into the environment, they don't degrade. They aren't broken down. They will last forever. And for that reason, they've been given the moniker forever chemicals. This doesn't just apply to the outdoor environment. It also applies to the internal environment of an organism's body. Because these PFAS chemicals can't be readily broken down and metabolized, because no organism has the enzymes to do it, these PFAS chemicals will accumulate in the organism's tissues. And to make matters worse, these PFAS compounds are toxic. They can do various negative things to the organisms that they get inside. They are carcinogenic. They can disrupt animal hormone expression, and PFAS chemicals are surfactants, so when they mix with water, they can drastically reduce the water's surface tension. If you're an organism that needs the surface tension of water to be sustained, like a vascular plant, being contaminated with PFAS chemicals can be deadly. From here, it's very easy to see how PFAS contamination in a river or a groundwater source can have catastrophic consequences for downstream forest ecologies. PFAS chemicals are being phased out of consumer goods because of this environmental threat, this health threat. But this doesn't remove the PFAS contaminants that are already out there. There's a variety of ways to purify water or soil that's contaminated with PFAS, but they're all pretty labor intensive and they can be expensive, and they're not super effective. You don't get anywhere close to 80 or 100% effectiveness. Hundreds of millions of dollars have been spent on research for an effective bioremediator that can break down and clean out PFAS. Alright, so besides being the longest introduction ever, this is all interesting and somewhat grim, but it brings us, finally, to today's news. In a heavily polluted New Jersey wetland, researchers found a strain of microbe that actually has the ability to break this strong carbon-fluorine bond, and thus initiate the breakdown of PFOA, or perfluorooctanoic acid, and PFOS, or perfluorooctane sulfonate, which I mentioned earlier. Essentially, this is a microbe that can naturally metabolize and break down at least 2 common PFAS chemicals. Dr. Shan Huang and Dr. Peter Jaffa of Princeton University's Department of Civil and Environmental Engineering wrote and published the paper on this fascinating Actinomicrobium species, known as strain A6. In normal conditions, this microbe would oxidize ammonium and turn it into nitrate, and dump the collected electrons on an iron atom. This process is called pheamox, with the Fe referring to iron, the am referring to ammonium, and the ox referring to the oxidation reaction. So you have this pheamox reaction. The researchers found that strain A6, when put in otherwise regular soil that had been contaminated with PFAS, the strain would perform the pheamox reactions, and in so doing, they would break the carbon-fluorine bond of the present PFAS chemicals. After 100 days, strain A6 was able to reduce PFAS levels in the soil by up to 60%, which is incredible. I mean, that's huge, and it's super surprising too, because researchers had largely written off the viability of a microbial remediation technique. All of the data so far had suggested that no organism had any enzyme remotely powerful enough or shaped appropriately enough to facilitate that kind of reaction. But this Actinomicrobium species of strain A6 has disproven that hunch. The authors of the study say, and I quote, "Given that the pheamox process has been reported in many environmental settings, as long as the pH is acidic and soils are iron-rich, the results presented here indicate that under the right conditions, the pheamox process could be stimulated to achieve PFAS biodegradation in contaminated sediments and groundwater systems." Unquote. So to summarize, this is big. It's really big news. PFAS contamination of soils, groundwater, rivers, and wetlands is a huge problem. The sheer scale of the contamination and its geographic distribution is staggering. And as it's always been one of the hardest compounds to remove, dealing with it can be very expensive. This discovery means that we potentially have a much cheaper and much more effective alternative. It means that we might have a powerful new tool for cleaning up polluted environments. I really hope that this kind of biotechnology can make it out of the lab, and to do so quickly, because a discovery like this is game-changing, and it's long overdue.